Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Akmal Bermola. Today I will explain you about the introduction and microbial source of handling microorganisms. Introduction. Microorganisms are a complex group of organisms. Microbial cells are thus distinct from the cell of animals and plants, which are unable to live alone in nature but can exist only as parts of multicellular organisms. Since microorganism cultures may be affected by viruses or become virulent as a consequence of evolution, they should be considered as potentially dangerous. Pathogenic microorganisms may gain access into the human body if a body surface with wounds or cuts is in direct contact with the culture. When dealing with microorganisms, hygienic precautions should be taken. Gloves with a protective coating should be worn. Microbial source. Microorganisms may be cultured on agar medium or on foods that they normally grow on, such as fruit. Those associated with certain types of environment, for example, bacteria in water. Those arising from microbial decomposition of various substrates associated with particular occupation. Those stemming from effective individual harboring a particular pathogen. Never culture microorganisms from potentially harmful sources like waste, human mucus, pus, or the face of people. Just use non-pathogenic microorganisms only.
Hi, today I'm gonna explain about inoculation of culture. In microbiology, the definition of inoculation is different from other common terms such as health, vaccines, and immunology. Inoculation in microbiology is the study of introducing microorganisms into environments where they will grow and reproduce. In other words, we can say that inoculation means introducing a certain substance into another substance. For example, inoculation is adding a certain type of nutrient or chemical into a suspension of bacteria. There are a few methods for inoculation, but I'm gonna go for strict plate method. The strict plate method is mostly used to isolate microorganisms, mostly from bacteria, from a mixed population into a pure culture. Before we proceed to the method, we have to be alert on aseptic technique. What is aseptic technique? Aseptic technique are basically a precautions that are very crucial to avoid the people that handling this activity from getting any infection. Before starting the inoculating method, ensure that all surfaces should be swept with a suitable disinfection before and after all operation, and give a sufficient time to allow for the disinfection process to occur. The procedure for aseptically transferring microorganism is we have to sterilize the inoculating loop. The inoculating loop is sterilized by passing it at an angle through the flame of a gas burner until the entire length of the wire becomes orange from the heat. In this way, all the contaminants on the wire are incinerated. Never lay down the loop once it is sterilized, or it may again become contaminated. Allow the loop to cool down a few seconds before contacting the inoculum. This step is important to avoid killing the microorganism. The mouth of all containers, tube, flasks, and bottles should be flamed after removing their caps and before putting in their replacement. Next is transferring the inoculum into a petri plate. The first one is we have to lift the edge of the lid just enough to insert the loop. This is to prevent contamination from spoiling the lid from outside. For example, either microorganism that might came in the lid, such as microorganism that came from the air. Next one is when we're removing the loop, we must immediately close the lid as fast as we can. This step is very important and also be applied to prevent contamination from outside. The final one is we must remember to re-sterilize the inoculating loop by placing it with alcohol or flame for at least 10 seconds. This step is very crucial to kill microorganisms that still exist and to make them fully destroyed. For the tapping and labeling, the first one that we must remember is to mark the base of the petri dish rather than the lid with a permanent felt pen or wax pencil. This is very important to prevent confusion if the base separate from the lid. The second one is to always keep the whole petri dish package in your hands and use adhesive tape to seal the dish. The last one is to prevent contamination or unintentional separation of the base and the lid. It is quite possible to seal in such a way that gases exchange is still allowed.
Okay, the next slide is incubation of microbial culture. What is incubator? Incubator is a device that used to grow and maintain microbiological culture or cell culture. Incubator maintain a optimal temperature, humidity and other conditions such as the carbon dioxide and oxygen that needed by the microbial to grow. The advantages of incubator is we can set the range of the temperature so it can reduce the possibility of culture being interfered with or accidentally discarded. The picture that I provide in the slide is the incubator that we normally see in the lab microbiology. It's not same with the incubator used for baby or for the egg. There are a few precautions in incubation microbial. The first one is before we want to inoculate microorganism, we need to incubate the petri plate with agar at 20 to 25 degrees Celsius for 2 to 3 days. It's to prevent any fungi growing. Fungi grow most successfully at lower temperature. As you may know, fungi is one of the most dangerous microorganisms that can cause the death if they can attack your respiratory. Number 2, do not incubate at human body temperature of above 30 degrees Celsius because we want to prevent the risk of culturing microbes that are pathogen to human. Number 3 is all inoculate plate must be taped before incubation. It's to ensure they cannot be opened accidentally. Do this by fixing with 2 or 4 short strips of adhesive tape at opposite edges of the dish. Do not seal completely as this may promote the growth of anaerobic pathogen. After inoculate, the lid and base of agar plate should be taped together with the same method that I mentioned before. It's for a protection from accidental or unauthorized opening during incubation. The picture shows the way you need to tap your petri plate. Last but not least, the plate that need to incubate must be upside down, which means the agar must be up, so that the condensation does not drip onto the plate and interfere with the developing microbe. After incubate, we are going to examination of microbial culture. There are few steps that you need to follow while during examining the microbial culture. The first one, sanitize your hand. Normally we use the sanitizer that have a 70% of alcohol because 70% is enough to kill or with the germ. Number 2, you need to wear gloves. Make sure your glove doesn't have any hole because we doesn't want any microbe to go near to your nail. Number 3, disinfect workplace area with alcohol before and after use. As I mentioned before, 70% alcohol is enough. Number 4, don't spit or open mouth while open the plate culture because to prevent the microbe from enter your mouth.
Hello everyone, my name is Muhammad Amza bin Muhammad Zahir, and for my part I will explain of how to dispose of unwanted culture and contaminated materials, handling a microbial spillage and also sterilization after microbial work. So let's take a look at the first one, which is the disposal of unwanted culture and contaminated materials. There are two common ways that we can do to dispose, which is the first one is we can immerse it in a disinfectant such as using the sodium hypochlorite, or the second is we can use the steam sterilizer, which is an autoclave. An autoclave is a machine that uses a steam under pressure to kill a harmful microorganism. With the autoclaving also, you can use to sterilize a contaminated apparatus. So here is the image of an autoclave. Next we go to the handling microbial spillage. There are few steps for this. The first is when the spillage happens, you must inform to your teacher or laboratory technician to deal with it. Second, do not touch with unprotected hand. You must be wearing a disposable glove. The third is covering the spillage area with a towel that has been soaked in a disinfectant, such as hypochlorite, and leave it for about 15 to 30 minutes. And finally, swap the towel into the container for a disposal purpose. The last one, we go to the sterilization after microbial work. After done your microbiological work, the bench must need to be swabbed with the disinfectant, such as here using a Virkon. Then before leaving the laboratory, do not forget to wash your hand with a water and soap. And for the drying purpose, using a disposable paper towel and throw it into a container with a lid. And that's all for our presentation. I hope you will really enjoy. Thank you very much.